Hi, and welcome back to our module on dissolved gases. This is Matt Ross again. Today we're going to be talking about dissolved gases in the real world. And I'm going to shrink myself a little. So the learning objectives for this video are that you will be able to calculate gas exchange velocity and understand methods for measuring K, or gas exchange velocity, in the field. You'll be able to use this understanding to describe the major controls on dissolved carbon dioxide, oxygen, methane, and other important gases in freshwaters and in soils. And you'll be able to describe how gas concentration in soils can supersaturate, which means that it goes above 100% of the concentration in air, and consequences of this supersaturation. So, in previous videos we've talked a lot about Henry's constant, so the K, the sort of gas solubility, but there's lots of other Ks when you're talking about dissolved gases, and one of them is is the Henry's, or the solubility of gas, which is Kh, like we've talked about earlier. Another control on how much gas will dissolve into a solution is the atmospheric pressure. And then the final one that we haven't talked about much yet is the gas exchange velocity, or little k. And that can be abbreviated with a capital K or a little k. It's kind of confusing. But here I'll try to be consistent by using the little k, lowercase k. And if you remember, Henry's constant is the sort of this number that varies between gases, and it tells you something about how likely they are to dissolve into solution. And that's sort of a constant that you can look up for a bunch of different gases, or you can calculate in a bunch of different ways. But in the environment, water's not perfectly still. It's not perfectly at the same temperature all the time. And so what we tend to want to calculate more than Henry's constant is we really need to calculate the gas exchange velocity, or this little k. And so that's just how much gas is being exchanged with the atmosphere. And this gets into an idea that Richard's going to cover more in a future video, but gas exchange is really a process that's at disequilibrium. So we don't really, it's not really an equilibrium process. And so we're going to talk a little bit about kinetics and reaction rates in our next set of videos. But it's important to know that the way we think about gas exchange is because it's at disequilibrium. And so, and that's true in rivers and lakes. That's definitely true in soils. Gas is at disequilibrium. And so Henry's law doesn't really allow us to do much on its own, because it sort of assumes equilibrium. And so we need to sort of adjust our approach. And Nick introduced this formula earlier, that the flux of a gas is related to the gas exchange rate, a little k here, times the gas in the atmosphere at equilibrium minus the gas concentration in the water. And most modern research, the first thing that they're trying to do is try to figure out what this gas exchange velocity is. So if we go back to the video I showed in the first lecture on introducing dissolved gases, this video, sort of like I said, it shows some areas that are white, and that's just areas with pretty high gas exchange velocity. That water is really turbulent. It's exchanging with the air, and it's incorporating a lot of air. And then areas that are sort of less white and a little more, not exchanging as much with the air, you might have less gas flux coming out because there's less sort of exchange with the air. And that would mean that there's, even in this single scene, this one image, the gas exchange velocity across this river is highly, highly variable. So you could have the gas exchange being something like, 10 in one part of the river, and then maybe something closer to 10,000 in another part, and that those higher numbers are going to mean higher exchange velocities, and the lower numbers mean, of course, lower exchange. And a big part of when you're thinking about what dissolved gases are in solution, is there a bunch of methane in the water? Is there CO2 in the water? Is dissolved oxygen higher than 100%? You're going to be thinking about how does K vary across cross section of a river? How does it vary in a lake? How does it vary as you go downstream in a river? And so this sort of, I think, movie just captures how dynamic this gas exchange velocity could be, both at a single place and across different moments in time. So we'll kind of keep going, but so finding gas exchange velocity is really not easy. It's something that is, it's not like a lot of the other things we talk about in class where we sort of solve the issue. Basically, people are still doing all kinds of work on how to better estimate gas exchange velocity. We do know it depends on some things that we've talked about in previous classes, but for one, gas solubility, gas exchange velocity is going to depend heavily on the temperature, and so you can see as you get higher, the higher temperatures, you have higher gas exchange velocities, and that's sort of very common across all gases really. And then another major control is that the gas exchange velocity here, they're saying K600, it's just normalized to a specific number, varies a lot with the energy in the system. So on the y-axis here we have gas exchange velocity, and on the x-axis we have the amount of energy in the system. And here that's some multiplicative of the slope of a stream. This is a stream figure, and the velocity of the water in that river. And they're just showing a big breakpoint. But what I want to draw your attention to, maybe with this pointer, is how variable this gas exchange velocity is. So it ranges from sort of like 0.5 meters per day to something on the order of 1,000. It can be even higher. So gas exchange velocity really varies a lot in real systems. And gas, sort of to simplify what those figures are showing, gas exchange velocity generally scales with turbulence, which turbulence is sort of a function of the slope of a stream, and if we're talking about a river, times the velocity of that water. Obviously, if you're in a lake, the controls aren't probably going to be slope and velocity. It's going to be something about the sort of movement of water in the lake, and that, of course, is the same for the ocean. And then it's also going to be a function of temperature. And if the internal production of a gas is greater than exchange, you will start to get systems where you have supersaturation. And supersaturation is this really important idea where we can see in this oxygen curve here that if the dissolved gas is higher than the concentration of oxygen in the air. So when you have oxygen saturation above 100, that means that this system has some algae or some macrophytes that are producing oxygen during the day, and that means that the system can supersaturate, but that only happens if the gas exchange with the air is not fast, so it can't sort of compensate. So oxygen gets built up during the day, and then at night it gets consumed because the algae turn off and they might get eaten, and there's respiration, and so you have this like what's called daily or diel variation in oxygen saturation. So here we're just kind of focused on general ideas of how do we estimate gas exchange velocity, and that's a huge area of research. Like I said, there's a lot of different ways to do it. I'll go through just a few of them, some of the more common ones. So one that Nick actually showed is you can just straight up measure it directly. You can measure the gas exchange velocity directly. This is going to work better in lakes than it is in rivers, but it's something you can do. You can use noble gas tracers, so we'll talk a little bit about how those studies work. You can stay awake for a long time, so you can use these daily oxygen curves to measure oxygen right as night falls, and then that decline in oxygen will tell you something about the sort of natural gas exchange rate. And you could use forward or empirical modeling, which is basically taking information about the system or a bunch of estimates of K so that you can estimate it based on a linear regression or something for your system. Or you can do a Bayesian modeling approach that we'll talk about a little bit. So this is just an image of Nick again, and this is the measure it directly system. So this is an area where the water's moving slow enough that they can actually put out these little caps, these collars, and in those collars they're going to measure gas concentration. I think here it's probably a methane study, maybe also a CO2 study. So they might be measuring methane gas flux, and they just put these collars out on top of the water. They know that the starting concentration of methane is really low in the atmosphere, and so then they measure the concentration as it builds up in these little containers every 30 minutes or every 15 minutes, and then they build a curve that shows how fast gas is being exchanged into the container. And they can do that for as long as the system's still at disequilibrium. And then once you get that rate, that's your sort of gas exchange velocity. This works really well in sort of lakes and slow-moving systems where you can actually directly measure the gas exchange coming out. And it's a really effective method for let's say the trace gases, so something like carbon dioxide or methane or nitrous oxide, other gases that are low in concentration in the atmosphere. Whereas doing this with dissolved oxygen, you already have so much oxygen in the atmosphere, getting the differential between oxygen in the container and the oxygen in the water would be really hard. We don't have sensors that are sort of accurate enough to do that. So this works better with sort of trace gas studies like the ones Nick was doing. So if you're in a faster moving system where you can't actually like put a collar out because it'll get sucked underwater or something because you're in a turbulent river, you might want to use a noble gas or a tracer injection. So you can add a stable gas like argon or propane. Propane's not a noble gas, but it's a very stable gas to a river reach at a known rate. And then you calculate the difference between the gas added and the gas concentration measured downstream. And that basically approximate the gas exchange velocity, but then you have to apply that gas exchange velocity of propane through some sort of empirical method to get the gas exchange velocity of a gas that you're interested in, like dissolved oxygen or CO2 or methane. It's a fairly straightforward method, sort of to show you more. Let's say this is a small river where we really want to measure gas exchange. What we would do is at the top of the river, we would get a propane tank like you would to have a grill. Oh, I need to move my camera. I'll move it down here. I'll just move over here. We'll add this propane at the top, just drip it into the stream basically with a nozzle. And then some distance downstream, we will take gas samples and measure the amount of propane that's coming out of the system. And then we can use the sort of tracer injection math where the concentration X, the concentration we measured downstream, is equal to the concentration we added times the exponential of K, so the gas exchange velocity, over that distance. So this is the river distance. And this is a really common method for like high gradient streams where you couldn't put out a collar like Nick was doing. And it's a really effective method for these streams to measure gas exchange velocity. And then the last part, of course, is that you need to convert propane or argon to oxygen based on sort of lookup tables basically about the relationship between gas exchange velocities across those different gases. So similar to the tracer idea, you can also use what I jokingly here called auto tracers, or you can stay up for a long time or use a sensor. And so this is the idea that if there's something in the system that's adding gas at a sort of known rate, and then all of a sudden that process gets shut off, like let's say photosynthesis, then we can go ahead and use that change in dissolved oxygen once photosynthesis shuts off, so once nighttime hits, to estimate dissolved or to estimate the gas exchange velocity. And you can kind of see this in this figure I showed in our first video that we watched. But you can see that there's daily variation in oxygen at all of our research sites. And that daily variation sort of goes to supersaturation, and then it goes back down below 100% and then back up. And when it's going up, that is a process that's the algae production of oxygen, and they're adding to it. You can see that it does supersaturate here, so the gas exchange rate is lower than the production rate of oxygen in this system. And then at night, when it shuts off, we see this steep decline, and we can basically use the velocity of this decline to calculate gas exchange velocity. And that's a really common method, especially now that you don't have to stay up all night measuring dissolved oxygen. Instead, we can just put sensors out in the stream like we did here. And so this is really common, and this is a really popular newer technique. It works best in streams that have a pretty steady diel curve. So you could see that if you had a flood, you might think that something happened to your gas exchange velocity. Indeed, it did. It changed. So you'd have to sort of like use the method during those times when you have these really clear daily curves, and then apply it to other time series. So that's another way. And then finally, we'll just look at this video again. You can use empirical modeling, and that's just saying like, I know a lot about a lot of other reaches that look exactly like this one, and I'm going to estimate gas exchange velocity just by describing this river reach. So let's say this reach has some slope and it has some width and area, and I'm just going to sort of estimate gas velocity. And this is best for sort of like really large scale studies where you're not going to feasibly be able to measure gas exchange velocity. And then finally, the sort of forefront of this is, again, what Alison Appling has been working on. And so this is sort of using these dissolved oxygen time series and Bayesian modeling to estimate gas exchange velocity based on the data itself. And so this is a really promising method because all you need is a dissolved oxygen time series, which we have a lot more of those because putting oxygen sensors in stream is getting cheaper and cheaper. And this, like I said earlier, allows you to calculate cool ecological metrics like gross primary production or ecosystem restoration. And all of this is reviewed a lot in this paper by Amber Olseth and Bob Hall, and they really show this really nice thing at the end on how you might estimate gas exchange velocity depending on your study objective and your study site. And so I won't walk through this, but you'll have access to these slides. But basically, if you're doing large regional stuff, then that empirical model approach would work well. If you have a really high gas exchange area, you might use a tracer gas addition. And then there's other ways that you can do it here that we've talked about. So a key idea here is that gas exchange velocity is a dominant control on gas concentration in streams, rivers, and lakes. It is controlled by temperature, turbulence, and the gas's solubility, or Henry's K. So a lot of what I've talked about so far is sort of with a freshwater focus, but let's think a little bit about soil gas exchange. So soil gas exchange depends a lot on how much water is in the system and other features of the sort of mechanics and porosity and other things. And I'll just talk a little bit about a research project I did early in my PhD. So this is just a time lapse of a Venetian marsh being flooded, which is a daily occurrence, of course, in a tidal marsh. And so what you see here, if we go back, is at the beginning of the video, you can see that only the lowest sites are still flooded. The higher sites, sort of that have all these sensors in them, I was measuring dissolved oxygen and CO2 and water pressure. Those aren't flooded yet. And so when these aren't flooded, the gas in these areas, they have free and open exchange with the air. So unless there's sort of micro pockets where they can't be exchanged, a lot of that dissolved oxygen that's in the soil can be exchanged with the air, and then any CO2 that's produced by the roots or root respiration or anything, or microbial respiration, that can also be somewhat exchanged with the air. But as soon as you have the flood come in, and this video was recorded over about 6 or 7 hours, it's just a time lapse. As soon as the water caps it off, now it's going to be harder for that gas to exchange freely with the air because it has this cap of water on top of it. And as you can see, that water came in, and there's not a lot of turbulence. That dirt that you see is actually, we were at the site, so we were mucking about adding a little turbidity plume there. And when the water sort of is flooding this, now gas exchange is slowed down. And the goal of this study was to figure out, can these plants overcome this flooding and actually provide enough dissolved oxygen into their roots even when they're completely flooded? Because plants, for a lot of reasons, want to keep their roots oxygenated. They want to have high oxygen concentrations in their roots. And we had two different plants here. You can't quite see them, but the one up that the mouse is scrolling over, was called Sarcocornia, and then the one below it was called Limonium. And this was an experiment where we were looking at the plant effects. So we had these same plants, and then in one site we cut them, we removed the plants with a paired site, and then in the other site we left the plants intact, and we put these little green cages around them to make sure that it was an isolated volume for measuring gas exchange. And so again, what we were focused on here was, can these plants maintain high concentrations of dissolved oxygen even when they're flooded? Oh, I'm going to go back into that. And what we found was really interesting. Oh yeah, and before I get into that, I just want to remind you of some basic biology and redox chemistry that we're going to get into a little bit later. But respiration converts, converting carbon usually to energy, produces CO2, and is most efficient when oxygen is around. So when you have soil respiration or anything like that, you consume oxygen. We'll talk a ton about this in the redox lectures. So in isolated environments that aren't connected with open air exchange, respiration consume all of the oxygen, and that has a bunch of negative consequences potentially for plants. So this can cause a cascade of biochemical redox reactions. If you've ever been to a marsh and you smell that rotten egg smell, that's the result of some of these redox reaction. So we'll talk a lot more about this, but hopefully that little reminder will make the next set of plots a little more understandable. So again, we had two plots, some that were limonium. So on the right here, this is a limonium plant, and on the bottom is the Sarcocornia plant. And if you look at their root differences, that was a main focus of the study. So now we'll watch just this little video, and basically I'll pause it to explain what's going on. In blue is the level of the tide. When it turns to this cyan color, that means that the marsh platform was flooded. So that means that this site had water over the top. And then the black and red lines here is the dissolved oxygen concentration. So you can see that in the limonium sites, both where they were cut and where they were intact, there's dissolved oxygen was always 0, even when it wasn't flooded. And so what that means is that basically lower in the soil profile, so these oxygen sensors were about 15 centimeters down, there's not ever really high enough dissolved oxygen concentration for us to measure. And if we look at the Sarcocornia sites, we can see that actually we do have dissolved oxygen concentrations that stay high, and they are they're correlated strongly with daily signals of sunlight coming in. And these blue black bars are nighttime when we don't think there was photosynthesis going on. And so you can see you have these like tidal variation, and you have dissolved oxygen variation sort of related to those tides. And the key idea here is that only Sarcocornia, where there was this one plant, was there enough oxygen pumped into the soil to maintain an oxic environment where you had oxygen concentrations around 10% of total air. So this is sort of not an oxic area. And where we cut the plants, there was no oxygen in the soil profile. So that's sort of this idea that these plants are pumping oxygen into the soil, and that we saw this really clear response here. And where we cut those plants, there was no response. And that was only true for Sarcocornia, and I think that's likely because of its root structures. You can see there's a lot of really fine roots, so it could probably pump more oxygen than limonium, which has just like one taproot. So we might not have put the oxygen sensor right next to that taproot. So this is a really important idea in marshes for sort of how do they maintain some amount of oxygen in their root profiles. But that is really also important. If we're not thinking about dissolved oxygen, we can think about dissolved carbon concentration. Sorry, I'm trying to get to the next scene. And basically that would be the inverse of that. So plants, when they respire and when microbes respire deep in the soil profile, if that CO2 that's produced from that respiration cannot be exchanged with the atmosphere, you can get really high concentrations of CO2 in soils. And that increases as the soils get wetter because of the same process we're talking about in the marsh, where when it's wet, it's harder for that gas to freely exchange with air. And so this is sort of supersaturation. Supersaturation of carbon dioxide is a critical part of how weathering happens and why weathering is usually faster where there's lots of plants. And that's because plants are sort of pumping CO2 into the rocks. And if they're a little bit wet, then that CO2 becomes highly supersaturated, much, much higher than the 450 or 420, depending on what you're talking about, parts per billion. Oh, no. So that's sort of the main. We got to the last slide before I accidentally hit something. So that's the main sort of part of this. And then, of course, when you have highly concentrated carbon dioxide, just a reminder of the carbonate lecture, you create carbonic acid, and so you lower the pH of the system, and that is why weathering rates are faster when you have these supersaturated soil profiles of carbon dioxide. So the key idea here is that both freshwaters and soils can have gases that supersaturate. This happens when the gas exchange velocity is much lower than the production rate of the gas. Similarly, gases can be completely depleted in local environments with little or no gas exchange, like we saw in those marshes where we cut the plants. Here's the final slides with the information, and hope you have a good day.